Okay. Welcome, everyone, to this Curator Conversations at the ROM. I just want to make sure, Ivo, if you can give me, raise your hand if you can hear me properly. Perfect. So hopefully everyone can hear me. This is a bit of a weird situation, of course, where we do presentations like this virtually now. But it might be ongoing for the foreseeable future. So hopefully we can make something really fun and interactive out of it too. My name is Sebastian Kvist. If you don't know me, I am the curator of invertebrates at the Royal Ontario Museum. And I am absolutely delighted that you could join us for today's curator conversation. This is a digital program that explores themes and subjects from ROM collections that are carried out alongside industry and research professionals and experts in various fields. Today's curator's conversation is a bit of a special one because it simultaneously celebrates the International Darwin Day. Darwin Day, of course, inspires people around the world to reflect and act on principles of intellectual bravery, perpetual curiosity, scientific thinking, and hunger for truth, as was embodied by the great Charles Darwin. I'd like to begin by acknowledging that the ROM sits on what has been the ancestral lands of the Wendat, the Haudenosaunee Confederacy, and the Anishinaabeg Nation, including the Mississaugas of the Credit First Nation, since time immemorial to today. And it is my absolute pleasure to welcome Professor Ivo Francischetti to this conversation. Professor Francischetti is an assistant professor at the Johns Hopkins University School of Medicine, and he focuses quite heavily on pathology there. His area of clinical expertise surrounds hematology, coagulation, and hematopathology. And you'll probably hear quite a bit more about these complex words throughout the talk today, in particular, as he focuses on coagulation and anticoagulation, perhaps. Professor Francischetti worked as a staff scientist at the National Institute of Allergy and Infectious Diseases at the National Institute of Health in the USA. He worked in areas of vascular biology, vector and molecular biology, including malaria. He applies these research areas and clinical expertise in coagulation, understanding of coagulation and anticoagulation, including blood thinners, of course, and hematopathology. And he studies the bone marrow and hematolymphoid neoplasms. Now, if you don't know what hematolymphoid neoplasms are, then join the club, because I didn't either. So I had to Google it, and I found out that hematolymphoid neoplasms is one of the several classifications of lymphomas. Professor Francischetti and my own research intersect, though, whereas I don't study lymphomas, we both have studied the function and the finding of anticoagulants or blood thinners in the saliva of various animals and how these can be applied to human medicine, but also in other animals around the world. And these blood thinners or anticoagulants will feature quite heavily in today's conversation. As we begin, though, if you've ever seen a presentation that I've given, you might know that there will be blood. I love that term, and quite often I like to warn the audience that there will be images showing blood. There will also be images showing a variety of animals, including snakes. And so I just want to warn about that. And actually, before I invite Professor Francischetti to the conversation, I was hoping that you might be able to put up the first slide here. And what I would like to do, first of all, is give everyone a bit of a sense of what we're talking about when we're talking about blood thinners or anticoagulants. What you're seeing on screen here is basically a standard coagulation cascade in you're gonna go back. There we go, perfect. So it's a standard coagulation cascade in vertebrate taxa. And what that means is if you accidentally cut yourself, or if you fall whilst playing soccer and you get a bit of a bruise, then your body sets in motion all kinds of reactions that eventually lead to the clotting of the blood. And this blood, this process happens so that your body does not lose this precious fluid that's inside of it. I think everyone has probably at some point felt clotting blood on their body because you probably bruised yourself or potentially cut yourself by accident, and you found that the blood starts flowing, and then eventually it actually coagulates or it turns into a clot. Well, what happens is your body has created all these kinds of different proteins that create this blood clot. And there are two main ways that, in particular, leeches, which is the focus of my research at the ROM, a way that they actually attack the different parts of the coagulation cascade. But in red here, you'll see the two different main endpoints of the coagulation cascade. These are the cross-linked platelets and cross-linked fibrin. And those two different sections are what the blood-feeding animals are actually trying to prevent from happening. And they do this by attacking this coagulation cascade at various points. If you can flip one slide ahead, Ivo, what you're seeing here are the various points in which leech anticoagulants attack this coagulation cascade. So what happens when the leech feeds is that it pumps you full of proteins that attack the coagulation cascade at various points, and then prevent eventually the cross-linking of platelets and the cross-linking of fibrin. And what this leads to is a prolonged bleeding in the site, so that the leeches or mosquitoes or sandflies or black flies can feed for extended periods without the blood clotting. That is what they're aiming to do. All this to say that the proteins available in nature that act as blood thinners are very, very diverse. And what we'll be talking about today are only some of them. So I just wanted to give an overarching view of these different anticoagulants, in the understanding that they are very, very diverse. There are lots of different proteins that act on lots of different parts of the coagulation cascade. But Professor Francischetti, it turns out that leeches, although they are some of the most renowned blood feeders, of course, and some of the animals that have been leveraged for human medicine, perhaps because of their blood feeding nature in the past, they're far from the only animals that feed on blood, and there are lots of arthropods and insects that actually do the same, right?
Yeah, that's right. Thanks, Sebastian, very much for the kind introduction, and also the kind invitation to take part of the Darwin's Day and the Curator conversation today. I'm very happy to be here and to chat with you all and your audience and all about the blood-feeding insects, ticks, vampire bats, in the context of how they find blood and how they feed on blood, and why that is important in the end for us, right, as hosts of some of the diseases that these animals may transmit. But we're going to be focusing mostly, as Sebastian was saying, on the mechanisms that help these animals to feed on blood. So very quickly, I just wanted to remind you that there are hundreds of different types of mosquitoes, in the world. They are spread all over, and what this slide is showing here is one mosquito, one species called Anopheles that transmit malaria, and here is the stomach or the midgut, to be more technical, of the animal. Of a mosquito, and this is another mosquito that transmits dengue or yellow fever, Aedes. It seems to be very similar, but in the end, the salivary composition of these mosquitoes is quite different. When you compare them, the proteins, and just to have for you to have an idea, there are almost 15,000 different species of blood-sucking arthropods in the world. So it's a pretty diverse number of mosquitoes, and also I wanted to remind you that ticks is another very fascinating creature. Because they are also spread all over and the world. And one of the important things that we are seeing here is that the weight of a tick really goes from 2 days feeding. You can see that is not much, then 7 days, and then 14 days. And the important thing here is that this is possible because of the saliva allows ticks to feed on blood for many, many days. There are many anticoagulants, a lot of other substances that block the immune system, etc. But it's because the saliva and the salivary gland that ticks can feed so successfully on the host. And I also like to remind you that not only invertebrates feed on blood, but also mammals like vampire bats. This is Desmodus rotundus, is a very efficient blood feeder. And as you know, it also a vector of human rabies. So it has some public health implications, animals that feed on blood, not only because of malaria, but rabies and Lyme disease, among many others.
So just to make sure that we're following and that I'm following as well, would you, the vast majority of blood feeders in the world are invertebrates, of course, but what's the diversity of blood feeding non-invertebrates in the world? I think that the vampire bat has 3 different species, and they're the only ones only non-invertebrates that feed exclusively on blood. Is that true?
Yeah, that's correct. Well, I think there are about more than 1,000 different species of bats. Most of them are feeding on fruits. So
Yep.
most of the bats that we see, flying in the evening, they are not, searching blood necessarily. And also geographically, they are more restricted. They are not everywhere. And among the 1,000 or more, there are only 3 that actually feed on blood. And the only diet that they have is blood. They cannot survive without feeding on
so
blood.
they need it desperately. Okay, so in thinking of that, but also in the spirit of Darwin, of course, and thinking about the evolution of blood feeding amongst different animals in the world, I know the different evolutionary adaptations that leeches have had to undergo in order to feed exclusively on blood. But what can you tell us a bit about some of the overarching evolutionary events that have happened in maybe in insects even that allow them to feed on blood?
Yeah. Well, if we, for instance, in bats, they have to deal with a lot of red cells because they are ingesting blood. I mean, I'm talking more about how they digest, because there are 2 things. One is how they face the host immune response and the host vascular system. The other thing that is how they can prevent the iron or the oxidative stress associated with the red cells, because they are ingesting a lot of red cells. So all these animals, they have in their digestive system mechanisms that prevent too much free radical production, which can be very deleterious for these animals. So they are able to manage that. Right, and in terms of the evolution for the anti-hemostatics, these animals have been feeding on blood for millions of years, right? Ticks more than maybe 250 to 400 million years. So they have evolved in their salivary gland. They recruited genes that, through a number of molecular mechanisms, mostly gene duplication, mutations, these genes, they remain, if you want, in the tissue, and they pass to the offsprings because it gives some evolutionary adaptation. So some of these genes, they are important because they lead to the production of proteins, in other words, anticoagulants, for instance. And so over millions and millions of years, these genes, they have evolved, and they become very specific and very potent. Otherwise, they wouldn't remain, part of the genetic information that these animals have. And that happens across the board, in bats, in mosquitoes, in ticks. It's a process of evolution, right?
Of course. And you raise a very good point there, actually. I mean, one of the things that you need to do in order to feed on blood, of course, is get to the blood, right? So you need special mouth parts that can cut skin or cut tissue in order to get to the blood. But then you have to deal with everything that's toxic about blood, iron toxicity being one of the major ones. And then what you also have to do is be able to deal with a lot of red blood cells or with a lot of matter coming into your body as well. And a lot, just like we saw with the ticks, a lot of blood feeding organisms, they expand exponentially when they feed. And one of the reasons supposedly is because they need to be able to feed for extended periods and take in a lot of blood during the few times in their lifetime that that the blood is readily available to them.
That's exactly right. It's as you said, it wouldn't be able to do the job if this was not a product of millions of years of evolution, right? Now you pointed to the one of you mentioned about the mouth parts, etc., right? So there is that's another area that is very interesting as well. For instance, this is in the lab, right? We have this very illustrative picture.
That's amazing.
Yeah, it's these are mosquitoes that are is bitten a volunteer in a lab setting. So this is all controlled, and you do that for several reasons. One of them even to feed mosquitoes. But the important thing here that I wanted to mention is that mosquitoes, when they feed on blood, it's like it's causing a little bit of injury. It's a micro injury. Sometimes it's imperceptible. But it's causing injury because it's piercing the skin, right? And how does that happen? Well, this is a schematic diagram of a mosquito. So this is what we call the proboscis. You think about it as a straw, for instance. And the interesting thing is that it has the mouth parts here of the proboscis, right? There is one area where the saliva is released, and then another area where blood is collected and ingested by mosquitoes, right? And every blood-sucking arthropod has some peculiar mechanisms to feed on blood. And this is an actual image of the proboscis, and you can see how sharp it is, right? And that's how it pierces the skin, and then the blood flows through this channel into the animal, and because it causes injury, this of course right, there's an injury here, then it promotes blood clot. Like when we cut our skin, for instance, blood clot is one of the mechanisms we have to prevent blood loss. And so what happens is that almost every salivary component from ticks and mosquitoes, they have one anti-clotting, one inhibitor of platelet aggregation that is also part of hemostasis, and one vasodilator that facilitates blood flow, right? So this is a diagram showing.
This is interesting to me because when we look at leeches, they seem to have this incredible arsenal of blood anticoagulants, basically, and we know that they attack the coagulation or the coagulation cascade, I should say, in several different parts, and for that they need several different proteins, and we've just scratched the surface on mapping these proteins in leeches, although some are already used in human medicine. But what you're saying here is that saliva of some arthropods seem to have only a single anticoagulant and a single platelet inhibitor and a single vasodilator.
Well, I would say that they have at least one anticoagulant, at least one platelet. It really depends on how diverse, because it depends on the mosquito you're talking about, the tick. There are hard ticks, there are soft ticks. The composition is really different among them. So I would be more. I would like. I think that to say at least would be most.
They need at least one, but they do need each of these in order to feed effectively on blood.
Well, if they have one or more, they will they are more effective feeding on blood. Yes.
Very good.
Yeah, some of the species of the mosquitoes that have not evolved for so many years, they are not very good feeders, right? And like soft ticks, they feed for minutes, for 30 minutes, while hard ticks, they feed for days, and that's because the composition is different of saliva, the composition of the saliva, I meant.
Yeah. Yeah, and that's what we normally see. You know, this is partly why this conversation is so appropriate to have on Darwin Day as well, is because, you know, there are so many different blood feeders in the world, and it turns out that blood feeding has evolved, you know, dozens of times throughout the evolutionary history of animals, and they each have their own way of counteracting blood clotting, which is very interesting that it's evolved sort of independently from each other, that these very, very effective proteins have evolved on several different occasions, which makes the study of these things so fascinating, I think.
Exactly, and they use different scaffolds to do the same job. Different types of proteins, they will, you know, they block the same molecule in our body. Yeah. And another thing that I think is interesting for the audience is how we collect, right, saliva from these animals. This is a tick, right, and you essentially you can you know, immobilize ticks in a tape, and then you add a compound called pilocarpine, doesn't matter, but that helps the secretion of the saliva, and then you get like very few, you know, little drops from ticks in each of these little capillary tubes, and then you can centrifuge and you collect your saliva. And the other way you can collect salivary gland is essentially you have your mosquitoes under the microscope. You literally dissect the glands, and you have these are the glands under the microscope containing all the proteins that the salivary gland contains. Yeah.
That's amazing. Okay, so of course, we can see on the screen now that mosquitoes and anticoagulants, you know, up until now, we've spoken a bit about we seem to have an understanding of the benefits of anticoagulants, and we'll get more into that, I think, as well. But of course, there's also detrimental effects of blood feeding organisms in the world. It's not that you know they're all good for human health or human medicine. They're not all used for anticoagulation. And one of the backsides, of course, is that these blood feeding organisms quite often spread diseases.
That's exactly right, Sebastian. That's the other side of blood feeding. Because these animals have been feeding on blood for millions of years, they also transmit diseases, right? Because some viruses or some parasites can live in a mosquito, and they can live in the salivary gland of these mosquitoes. So when they feed on blood in humans, they may transmit disease, and for that reason, they cause a lot of death around the world. You would be surprised, but mosquitoes is the number one killer among animals, and this is mostly because they transmit diseases like malaria, right, and Chagas disease and sleeping sickness, etc. And then we have the others, humans. Snake venoms, about 50,000 per year, compared to mosquitoes is not that many. That mosquitoes, you know, 725,000 people dying. And then we have the others. Just an example, you know, it's very much advertised, but sharks kill 10 people per. That, I mean, of course, it's unfortunate, of course, but it comparatively is much lower than mosquitoes.
Yeah. And I think, you know, the since you're talking about the detrimental effects of being bit by blood feeding animals, and also the well, I guess that the benefits might outweigh those risks, but it seems as though, because blood feeding animals, and particularly mosquitoes, have this detrimental effect of spreading malaria and killing so many people, but also other animals, of course, every year, that the positive sides of finding anticoagulants that can help human medicine are quite diminished there. But I still would like to talk a bit about how blood thinners or anticoagulants that are used by animals in nature, how those can actually help humans in therapeutics and in medicinal uses. We know that leeches, for example, which is, again, one of the main research topics in my research lab at the ROM, we focus on trying to understand the diversity of anticoagulants in leeches. And we know that leeches possess what we think is the strongest or the most potent blood thinner, naturally occurring blood thinner in the world, called Hiridin. And it is synthesized and provided to patients that are at risk of stroke, Um, in some cases, to try to heal hernias or heal thrombosis under the skin, and it's used sort of in topological creams and in pill format. Are there any other animals that have these therapeutic potential?
Yeah, well, this is a very good point. Hirudin from leeches is the classical example of maybe one of the first molecules from blood-feeding animals that end up being used for humans.
Yeah.
But there are a number of other molecules that have been used as a prototype, in other words, as a template to create new therapeutics, or that are being used as drugs out there. There are many that are being used anecdotally that are not approved by governmental agencies like the FDA. And so it would be, we would be talking here for much longer. So I just chose three um, 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 examples of drugs that have been that are out there in the market, approved by the FDA, that were developed based on components from blood-sucking insects, or from particularly snake venoms, for instance, right?
Yeah, that's really interesting. Yeah.
So one of them, we all have heard about heart attack, angina, and coronary angioplasty. One of the benefits of the snake venom was that the venoms, they have some inhibitors of platelet aggregation, and platelets are very important because they are essential components of the thrombosis, right? And there is one of several molecules actually that prevents thrombus formation because they block platelet aggregation. That as I said, is a critical component of hemostasis, of thrombosis. And one of these molecules, there are two different types. They are being used for particularly for coronary angioplasty in some settings, not so common, but they are used, or for the treatment of angina. Nowadays, there are many other anticoagulants and the platelets that are being used, but this has been approved by the FDA at one point for these indications. So cardiovascular disease is one target for these drugs. The other very important disease is hypertension, and there is a snake venom called from Brothrops that a peptide was isolated, and it was used as a prototype to develop a small molecule that lower blood pressure. Okay, and has been a very, very useful and very effective drug for the treatment of a lot of patients with hypertension. Remember,
of course, the snake uses this in a very different way, of course, right? This is I think, a classic example of when humans have co-opted a protein that has been designed for something else, and use it for our own benefit, which is very, very interesting and quite smart of humans to do, because of course the snake is using it to kill its prey, to bring down the blood pressure inside of its prey so that it eventually passes on, of course. And so humans have co-opted that and to be used in human medicine.
That's exactly right. And the other thing is the discovery of this molecule happened more than maybe 50 years ago, when the investigators, the researchers realized that dogs, when bitten, as you said, had very low blood pressure. So they said, well, if there is low blood pressure, is because after being bitten by a snake, is because there's something causing low blood pressure, and we can perhaps identify this component and then develop this component as a drug that could help to treat patients with hypertension. So that's what was done. Exactly as you said, it was an observation that dogs, they have a very low blood pressure, and it became this drug called captopril, and many other different types of angiotensin converter enzyme inhibitors. Yeah, and a third one is another component that was identified in the saliva of the Gila monster. And this molecule helps to control the sugar in the blood of patients with diabetes, and that seems to be an effective way to that patients can be treated besides using insulin in patients with diabetes.
Yeah.
So these are just the three most, I would say, illustrative examples of molecules derived from saliva, from exogenous secretion. Yeah, it's very interesting. And since we have 15,000 different species of blood-sucking insects, likely there are many other potential therapeutics hidden in saliva of these animals.
Yeah, this was going to be one of my questions as well. And again, I'm only thinking of the sort of small research program that I conduct as well. Is we're just scratching the surface for finding therapeutic drugs inside of animals, whether it be anticoagulants or something else. You sort of answered it already, but do you have any sense of what is undiscovered out there and what it could potentially be used for? Do you have any inside information from Johns Hopkins or from your own lab that you could tell us about before the paper's published?
Not, I don't have a specific potential new drugs coming out of the. We have some interesting anticoagulants that are inhibiting the extrinsic pathway of the coagulation cascade. We have published a lot of papers about it. It has the potential, to be used as an anticoagulant. The only thing I would say the problem with these molecules sometimes is that they reach what we call a valley where it needs to have an additional step of development, because one thing is to discover an anticoagulant in the lab. The other thing is to bring that anticoagulant to the public, to become, and that's a very long process, because you need to know, after you discover that it's an anticoagulant, it's a blood thinner, you need to know that, okay, this molecule is not toxic to animals. It lasts, the effect last for at least 1 or 2 days, so you don't have to inject every 30 minutes, right? So there's a lot of things called pharmacokinetics, pharmacodynamics that we need to know, and not too many labs do that. The labs that work on research, they don't develop.
Right.
So,
no, there is exactly, and that's what we're finding. That's what we're finding as well. The basic research, the step from basic research to applied research is quite heavy. That's one of the reasons that I really wanted to invite you on here as well as the Darwin Day speaker, because you might have a bit more insight into the application side of things, and it's an important factor to state that finding an anticoagulant to the point where it becomes a drug, that step there is enormous.
It's a very long process. There are I know companies that devoted a lot of resources just to do that. There are maybe few that are still working on today, but it's not, unfortunately, something that is easily done. But to give you an idea, I would compare the hidden secrets of these animals in terms of what we might be able to find is like comparing the tip of an iceberg and the true iceberg, right? The iceberg, we just see the little tip, and behind that little tip we have a mass of ice that is I would think is what we don't know.
Yeah,
and the tip of the iceberg is what we know.
Yeah.
So there is a huge potential, I think, provided that these molecules, once discovered, then can go to a next step.
Yeah.
That is, as I said, there is a big. It's very difficult, unfortunately.
Before, and I think we should probably move on to the questions section pretty soon. But before we do, when we spoke the other day, we sort of tried to come up with a couple of different areas that would be really interesting for people to hear about. And one of those areas is quite topical. Now, it's important to say that this doesn't include any information about drugs. It doesn't include any information about anticoagulants or any sort of drugs that can be gotten from snake venoms. But there is a connection, and because you're a pathologist and a hematologist, I have to ask you this. There is a connection between corona or COVID-19 and thrombosis. Can
Oh
you
yeah,
explain that?
yeah, definitely. Yeah, it's a very, very good point. What happens is that in viral infection, they trigger a immune response that is called the innate immune response. And when this response has several components, and one of the components is the coagulation cascade. The coagulation cascade can be regarded as a mechanism of defense, and there's a crosstalk between the coagulation cascade and the immune system, particularly the innate immune response. The problem with COVID is that it affects the endothelial cells, and it activates the so-called intrinsic pathway by activating neutrophils, and these are events that are procoagulant. It stimulates the coagulation system through different ways, either directly, as I said, activating neutrophils, or because the endothelial cells that are our vessels, if they are affected by the virus, then the anticoagulant function of the endothelial cells are lost. I don't want to be too much technical here, but coronavirus, it increases the procoagulant tonus, the procoagulant status of our body in different ways.
Okay,
And
so your body could, or it has been found in some patients that your body is more prone to clotting the blood when infected with COVID-19?
well, that's the reason why they have thrombosis.
Yeah.
And the reason why that happens is, at least in part, because of the neutrophils. The neutrophils, when they release some sort of nuclei components called nets, and these nets, they activate the coagulation cascade. Right. And that can be very dangerous.
Yep.
Right. And on top of that, as I said, the endothelium, our vessels are being affected by almost like an inflammation of the vessels. Right. Yeah. So all these things combined make it very severe and difficult to treat, because it's too intense for some patients more than others. Why some patients develop it more than others, I think, is still an open question. Yep. But it's the basis of the disease, at least in terms of vascular biology, is coagulation activation. Yeah.
Yeah, that's really interesting. Obviously, very topical, and there's a lot more research that's going to be happening on Corona and COVID, and possibly when it comes to thrombosis as well in the next coming decades, certainly. And here's hoping that we can counteract COVID first, and then try to understand everything that we can about it.
Yeah, I think that that's the future in COVID, to try to understand the disease so that we can have better therapeutics, right? Hopefully the vaccine will be effective, so we don't need to have so many, but yeah, that's definitely the way to go, is learning the physiopathology of the disease, and then we can target the mechanisms. Yeah.
That's great. That's great. Thank you so much for that, Ivo. I think
sure.
We have about 15 to 20 minutes for questions. And so I just want to direct everyone's attention to the Q&A right at the bottom of the screen. If you click Q&A there, that's the place where you can ask myself or Ivo questions that relate to blood feeding or anticoagulants, etc. So if you've posted the questions in the chat, I think that Aaron very kindly has tried to direct you towards the Q&A, but the Q&A should be at the bottom of your screen. If you have any questions, just let us know. I have tons of questions for you, Ivo, but I think we should go to the audience's questions potentially. And the first question that came up here is, if mosquitoes are so dangerous, would it be beneficial to take steps towards making them go extinct? Now, this might be a question that we can answer together, but I'll let you take a first stab at that.
This is a good question. I don't know if extinction of a species is the best way to deal with it, right? For instance, malaria, it was endemic. In other words, was very frequent in many countries, like in Italy, for instance, right? And in that particular area, mosquitoes were extinguished they were decimated by different use of chemicals and etc. So it's likely in some areas there is a benefit for killing all the mosquitoes that are transmitting diseases, especially in areas that are very endemic. But I don't know if, because the ecosystem it has to be in equilibrium. So mosquitoes have a role in the ecosystem. They are they are useful as food for other animals as well.
And I think you're absolutely right, and that's the standpoint that I would take as well. Like, they have a role in the ecosystem, and they are incredibly important, and they're so numerous and abundant throughout the world that actually, trying to kill off all the mosquitoes would have very detrimental effects on ecosystems. One of the problems, of course, is killing off mosquitoes alone, or killing off, Anopheles or Aedes, the species that carry malaria effectively. Killing off only those and no other mosquitoes would be very, very difficult, but also killing mosquitoes in general would be detrimental to the entire ecosystem. In fact, I would argue that it would incur so much more damage to humans than malaria perhaps does, because all the organisms that rely on mosquitoes would also die out. In biology, we call this a bottom-up effect. Where the prey of certain animals become decimated, therefore the predator themselves become decimated, and then on top of that, there's the predators of the predators become decimated, the predators of the predators of the predators become decimated. So it would destroy an ecosystem effectively, I think.
Yeah.
The second question here relates to the excess of irons, and the question is, I'd be interested to hear how the bat deals with excess iron, as I know someone who requires monthly transfusions due to no red blood cell production, called diamond black fan anemia, apparently, if you know that term, Ivo. Yeah, iron buildup is a problem. I'm not entirely sure how bats deal with iron buildup in particular.
Yeah, but I know that for mosquitoes, there has been research on that, and they have some enzymes, some antioxidant enzymes that can get rid of of iron. In bats, I am not sure if the same research has been done. Right. I'm not familiar with.
Some similar studies have been done in leeches, and they seem to be able to contain the blood or contain the hemoglobin quite effectively, or the iron atoms inside of the hemoglobin, and they basically put them inside of a cocoon, and then they excrete the iron quite effectively, actually.
Yeah, the same. I mean, even small parasites like Plasmodium. We're talking about a very small. It has to deal with iron as well. With hemozoin is one of the aggregate iron, the hemoglobin. Right. To do that, yeah.
I hope that answered the question out there. The next question is, how is the compound from which exenatide, so that's one of the drugs, I believe, the compound from which exenatide has been formulated, used by the Gila monster? How does it affect the creature that comes into contact with the Gila monster saliva? That's a good question, yeah.
Well, so what happened is this: our blood sugar in our blood, is very important for all the functions that we have. So if you have a animal that produces very low sugar levels, that affects the ability of the prey to escape, because the sugar is the main source of energy that we have, right? Like when you start to run, it's the sugar that you have in your muscles, the glucose, that helps you to start running, and then the metabolism, like the fat metabolism, protein metabolism, it's later on, but you have to have some glucose, for that. So likely these animals, they found a way to facilitate hunting the prey by lowering the sugar levels of the prey, and that makes it easier for them to chase the prey. It's interesting that they found these mechanisms as opposed to finding an anticoagulant, right?
That's right.
So it has to do likely with the type of prey they have and the way the genes were recruited in the salivary gland. But essentially, that's what it's trying to do. Decrease the energy levels by decreasing the amount of sugar that you have.
And of course, there are some snakes that effectively, have such powerful anticoagulants that a bite will make the animal, well, it will create internal thrombosis, and basically that the animal will die from just internal bleeding, and presumably they're injecting an anticoagulant then.
Yeah. That's right. Sometimes they have, there are some paradoxical effects. You see thrombosis, in a vessel, and you see bleeding in another vessel. And it really depends because it's so complex. Sometimes you have an enzyme that, activates the coagulation cascade, and it consumes the coagulation factors that leads to bleeding. But sometimes you can have a protein, an enzyme, a metalloprotease that we call that can promote coagulation by indirect mechanisms. So,
very good. So the next question, I hope that answered the question. Next question is, how do you isolate and identify the proteins of interest? And perhaps we can come at this again from both points of view.
Yeah. Well, this is a very interesting. I mean, it really depends where you are in 10 years, because what we used to do 20, 30 years ago, we used to collect saliva to dissect salivary gland. It was really very time consuming. And then you have something called the protein chromatography, where you get all that salivary gland, all the saliva, and then you load in a small column that essentially separates all the components. And then you have to use a technique called Edman degradation that you get to know what is the composition of but it was really very time consuming. Nowadays, maybe for the last 10 years at least, it completely changed how we study the saliva, right? We have with a little bit of saliva, very few salivary glands. You can, using some techniques in the lab, using particular enzyme called the reverse transcriptase, you can amplify the sequences, the genetic information that you have for that particular animal, that particular salivary gland. Maybe you have 10 mosquitoes, and you can, with 10 mosquitoes only, using this technique, you can amplify the information, and then you clone or you sequence all these genes. And there are a lot of different techniques in the lab, bioinformatic, that will classify, with a lot of computer power, can classify the types of molecules that you have. So essentially, with very few glands today, you can have a pretty good idea of what that particular animal is producing in their salivary glands.
And I think you're absolutely right. And this is something, we're lucky that we're doing research in 2020, 2021. Liquid chromatography is not something that we do in the lab either. But like you said, now it's directly extracting the RNA and DNA that are in salivary glands, and then directly sequencing in a massive way every single protein that is expressed in the salivary glands, and so that becomes the baseline for understanding the proteins. And then you of course have to use highly sophisticated bioinformatics to try to tease out what's actually in the saliva and what's what might be a contamination, or what these different proteins actually, what the function of the different proteins are, etc.
Yeah, and I think that the knowledge that we have today is grew exponentially, because of massive sequencing.
That's right. Yeah, I think it's one of the major breakthroughs that humans have come up with in the last, I'd say, 10 years is next generation sequencing, or being able to sequence, parallel sequencing massively, I think.
Yeah, and just one point: once you have your sequences, you have to express the protein. In other words, you get that particular clone that we call our sequence of interest, and you introduce in a bacteria. This is recombinant expression that we call in the lab, and the bacteria will produce it for you. And so that's how we can purify and collect, large amounts of the salivary components.
Yeah, that's
of interest.
Um, okay, so I think we have time for two more questions here. So we have two more that are on screen right now. I'll do the last two, and then we'll see if there are any other anything else. I have a couple of last announcements as well, but we'll do two more questions if that's okay with you, Ivo.
Sure.
So the next question is: can the drugs for the three diseases that you highlighted, in particular for the snake venom, can they be produced solely synthetically?
For the three, so
I believe these were the
yes, they can be. Actually, the ones from the snake venoms, they're all. See, I didn't have enough time, but oftentimes what happened is this: you have a protein from the snake venom, but then the industry and the chemists in the industry, they modify that molecule that I call a prototype. So it's based on that prototype from snake venom that you create something else. So most of these proteins, they are synthetic components modified from the natural molecule, but it's not exactly the same. I'm not sure about the exendin 4, the one from the Gila monster, if this is exactly the same or not. But the one for treatment of hypertension that I mentioned, this is for sure is a modified molecule that you can even take by mouth. And the one for cardiovascular diseases also, they have been modified based on, as I said, a prototype isolated from the snake venom. And so that's why it takes a lot of time and resources. And the other thing is a lot of these molecules,
in the passing, but that step is also time consuming and very, very, it takes a lot of time and money to create that step.
A lot of time and money, and it's not, and the chances that it will fail exist. So it's not every molecule that you find that has a function, that works in animal models that work in humans. It's a very, very complicated process.
Okay, let's do the last question here. Are there insects or animals that, and this is a very, very good question, it's interesting, I'll be interested to see what you think of this, Ivo. Are there insects or animals that have a coagulating effect instead of anticoagulation, and that have been found to be beneficial in any way?
Have a coagulation
so instead
effect?
I think what the question means is that are there any animals that instead of introducing anticoagulants into the body that introduce procoagulants to clot the blood instead of anti-clot it?
Oh yeah, definitely. Yeah, definitely. There are many. Oh, this is another area that is very interesting because what happens is this: if you have a procoagulant is usually an enzyme. What is an enzyme? Enzyme is a protein that converts something to something else.
Yeah.
And if you introduce an enzyme that is procoagulant, it means that you are going to consume all the clotting factors. If there are too many, like snake venoms are notorious for having procoagulants. So it activates the coagulation cascade, and then with too much coagulation, you have a consumption of all the clotting factors, and in the end, you have hemorrhage because you don't have clot anymore. You don't have coagulation factors anymore to do the job if you need. So that's why a lot of patients, when they are bitten by some types of snake venoms that we call hemotoxic, they end up bleeding. So they have thrombosis sometimes, but they end up having bleeding. Thrombosis because there is coagulation activation.
Yep.
But at the same time, there's so much coagulation activation that the clotting factors are consumed and they bleed. So, on top of that, these enzymes, they can destroy the tissue that helps bleeding, right?
Yeah,
if you don't have something to prevent bleeding, if the tissue is being damaged by the proteases of the venom. So yeah, this is a well-known, very well known. Yeah, it happens a lot in snake venoms.
Yeah, and that was my immediate thought when I read the question as well. I think I've heard a lot about procoagulants in snake venoms, and of course, I mean, different snakes have different ways of killing their prey. Introducing anticoagulants can be one way, but producing procoagulants, just like you said, might allow the animal to bleed out, but it can also clot the blood so that blood doesn't get from the heart to the different extremities, which is an effective way of killing someone as well.
Yeah, and they're very potent, very effective, and kill very quickly, relatively quickly. Yeah.
Professor Francischetti, thank you so much for being with us here at the ROM for the Curator Conversations. I would like to take a second to thank the sponsors of the Curator Conversation, TD Bank, for their ongoing support for the program. I want to thank everyone for joining us. We hope to see you again for the next Curator Conversation, which is on February 17th. And I'd like to remind everyone that details of all upcoming ROM at Home online programs can be found on the ROM's website and on our social media channels. So please join me in thanking Professor Francischetti for taking the time out to talk to Toronto, especially when we're in a pandemic and everyone is working from different places. Some from home, some from isolated labs, and some from their apartments. So thank you so much, Ivo, for being part of this.
Thank you, Sebastian. It was a real pleasure. I really enjoyed to chat with you, and thank you everyone at ROM and the audience for the questions. It was really very nice. Appreciate it.
Thank you, and thank you everyone for your ongoing support for the ROM. Here's hoping that we can be up and running in the very near future. I hope that everyone stays safe and sound out there. Take care and stay warm in Toronto or wherever you are. Thank you so much, everyone.
Bye, everyone.